and welcome to another edition of the Science Education Series. Today, I am here with Dr. Jim Farmar, who is the principal scientist here at BioCat. Thanks for being with us today, Jim.
Thank you very much for the invitation, Carolyn.
Absolutely. So I'm just going to go ahead and get started. So first, can you give us a brief overview of your background and how it led you to BioCat?
Happy to. Arguably, it all started when I went to Brown University for my doctorate in organic chemistry. One of my projects there was to make a termite chemical that termites make without thinking, but I had to do a lot of thinking. And that was my first association with a biomolecule, which you'll see as a theme throughout our conversation. After Brown, I decided to go to Rockefeller University for a couple of postdoc years, and there I got to work with some more biomolecules, in particular proteins, and what happens to them during the aging process. From Rockefeller University, I decided to go to a biotech startup, again in Manhattan. And this was a company called Imclone Systems, and they worked with a variety of different projects, most of which had me analyzing biomolecules, and in some cases synthesizing biomolecules, either as short bits of DNA called oligonucleotides, or short bits of proteins known as peptides. Next, I went to the New York Blood Center, which is another research institute in Manhattan. And there I got my first taste of liquid chromatography, mass spectrometry. I'll say LCMS going forward after this. There I got to look at proteins, oligonucleotides, and peptides using the LCMS and a bunch of other analytical techniques. From the New York Blood Center, I went down to the University of Virginia at a mass spectrometry lab. And there I built on my skills, again, looking at more biomolecules. Before coming to BioCat, I spent some time over at Merck in the Shenandoah Valley. And then finally, I got a chance to come here to BioCat, and I'm working again on a biomolecule called enzymes. And that's what I've been doing for the last 3+ years here, using LCMS to look at the products that come out of enzyme activities, particularly the enzymes we use for dietary supplements.
Great. So could you talk to us about how you use LCMS here at BioCat, and could you walk us through a few of the steps that it takes?
Absolutely.
Great.
So why do we measure the activities of our enzymes? We have HPLCs with non-MS detectors in addition to our LCMS. The HPLCs allow us to identify and quantify the amino acids, the fatty acids, mono and disaccharides, which are the major products of our proteases, lipases, and oligosaccharides. I'll give you an example using proteases. Proteases digest proteins, and there's two types of proteases. One cuts in the middle of the protein, and the products that it produces are peptides, and so short bits of protein. The other type is kind of like a Pac-Man, and it starts from one end or another, and it literally chews off individual components of the proteins, and those components are amino acids.
All right.
The reason amino acids are important is they are then transported into your bloodstream, and they are the fundamental building blocks of protein manufacture within the body. So the example I'll give you is, what if you wanted to have a better enzyme or a better enzyme blend? If we wanted to compare how well, say, protease A digest a meal versus protease B, we measure the amount of amino acids and peptides that are released as a result of these enzyme activities. So as I mentioned before, we can use HPLC to quantify the amino acids, but we couldn't identify or quantify the amount of peptides produced by the proteases. With the addition of LCMS to the R&D laboratory several years ago, we were able to see much more of what our proteases are doing, and as well as all the other enzymes we work with here at BioCat. Knowing the peptides that each protease produces is very important. So the aforementioned HPLCs will give you a good idea of how well they're producing amino acids, but they won't tell you how well or what type of peptides the protease blends and mixtures are producing. That's what LCMS can do. In some cases, for some people, certain peptides that are, if you will, left over from digestion cause gastrointestinal distress. And a good example are gluten proteins, which produce peptides that cause problems. So if we could come up with a blend of enzymes or one particular type of enzyme that would totally digest the problematic peptides, that's going to be beneficial to people who have wheat or gluten sensitivities.
Sure.
We had a project a couple of years back that looked at an enzyme called inulinase, or sometimes now referred to as fructinase. And what it does is it digests fructans or inulins. And what we found by using LCMS is that it overwhelmingly produced mono and disaccharides, in particular fructose, glucose, and sucrose. And we could figure that out using the LCMS and looking at an assay. When we use the same assay to look at another oligosaccharide, this one being xylanase, we found a totally different result. It produced monosaccharides and disaccharides, but it also produced something else called ZOSes, which is short for xylo oligosaccharide. The reason why this is important is they have been shown to be very important as a prebiotic in terms of keeping people's general gastrointestinal health. So the LCMS has plenty of benefits in terms of other things that we've long needed here, which is we're able to characterize the protein sequence of our enzymes to better understand how similar they are to other proteins and other enzymes, and what other proteins or enzymes might be present in our enzyme blends.